Hello everyone. This is a short video for the methods of biology which will help in your practical, in your MSc dissertation work, in PhD work also, for experiment and for knowing which instrument or which technique is used for which application. Okay, so in this short video we'll know some kind of technique with their application to apply in your PhD work or in your experimental work. Okay, then methods in biology. First of all, this is First of all, this is the techniques in left side and the application is in right side. Okay. First, agarose gel electrophoresis. What is agar? Why the technique agarose gel electrophoresis is done? Okay. The application of this technique is to determine the molecular weight of DNA. Okay. By this technique, we can determine the molecular weight of what? Molecular weight of DNA. Okay. The larger molecular weight of DNA can move slower than the smallest molecular weight of DNA. Okay, smallest molecular weight of DNA can move faster in agarose gel electrophoresis plate. Okay, by applying this, we can determine the molecular weight because by the molecular in molecular weight, the what is the which is the larger molecular weight and which is the smaller molecular weight can be differentiated or can be recognized. Okay, then AFLP, that is amplified fragment length polymorphism. Okay, by this technique uh, we can determine the parental DNA analysis. Okay, that is a molecular marker. Okay, that is a molecular marker. Then what is the molecular marker? We can also set this genetic marker. Okay, then the molecular marker are used in molecular biology, biotechnology also. Okay, to identify a particular sequence of DNA. Okay, we have a quantity of DNA. Okay, we don't know which DNA, whatever DNA it is. Okay, so, for found our desired DNA, we apply the AFLP, that is amplified fragment length polymorphism. For what? For identify a particular sequence of DNA in a pool of unknown DNA. We don't know whatever the DNA is, but by applying the AFLP, we can analyze the DNA, parental DNA. Okay, we can identify the parental DNA. Then affinity chromatography. Affinity chromatography. What is affinity chromatography? The for the application, why affinity chromatography is used for the application of mRNA purification. Okay, and then three types of RNA: mRNA, tRNA, and rRNA. So we can identify the first, the identification of mRNA is occurred. Then after that, the identify, not identification. First, the extraction of mRNA is occurred. Then purification of mRNA is occurred by the application, by the technique affinity chromatography. Okay, so then cloning for what is the application of cloning? For foreign gene transfer. Okay, we can also call it cloning vector. Okay, so vector, see, this is cloning cloning vector also. For then the cloning vector is what? Vector is act as a vehicle to transfer a gene from an organism to another organism. Okay, Vector is act like a what? Vehicle. Okay. Then for engine transfer, it can transfer pathogen, it can transfer disease. Okay. And then this is the foreign gene transfer method, cloning. Okay. Then CHIP, chip, chromatin immunoprecipitation. Okay. This technique is used for chromosomal deletion or addition. We studied about chromosomal aberration, right, in genetics. Okay, that in chromosomal aberration, the chromosomal deletion occur, chromosomal addition occur. Okay, so we can identify, or we can analyze the which part of chromosome is deleted, or which part extra addition of chromosome. Okay, it can identify by the application of chip, chromatin immunoprecipitation. Then CD, circular dichromism. Okay, circular dichromism. It is, now what's the application? There is secondary structure of protein. Okay, there are different type of techniques for primary structure of protein, for secondary structure of protein, for tertiary structure of protein. Okay, there are different techniques. So in secondary structure, so with the help of CD or circular dichromism, we can see the secondary structure of protein. Okay, then chromatography. Okay, what is chromatography? For protein or RNA purification. Why chromatography is used for protein or RNA purification? First of all, we extract a protein or we extract RNA, then we purified it by the application of chromatography. There are various kind of chromatography. Okay, paper chromatography, ion exchange chromatography. There are various types of chromatography we used in our experimental use. Okay, then confocal microscopy. Microscopy also are various types. Okay, confocal microscopy, phase contrast microscopy, electron microscopy. Okay, there are various type of microscopy. But in confocal microscopy, what is the application? 3D image of tissue and cell. Okay, 3D image of tissue and cell by the confocal microscopy. And then DNA microarray for the application gene expression analysis. Okay, how gene expression analysis um, can be identified by the DNA microarray. Okay, then ELISA. What is ELISA? Enzyme linked immunosorbent assay. Okay, then why ELISA test is done? You know, and to see if you have been exposed to bacteria, viruses, or other agents that cause infection. Okay, for that we do ELISA test. Okay, for quantitative or qualitative analysis. That is how the if it have been exposed to bacteria or virus or other agents that cause infection. Okay, then we then FISH technique. What is FISH technique? What is FISH? Fluorescence in situ hybridization. Okay. Then it is a in vivo DNA or gene expression. Okay. What is in vivo and what is in vitro? Okay. These two types are the type in vivo and in vitro. In vivo means two types are the research related terms, in vivo and in vitro. Okay, then in vivo means research done on a living organism. Okay, on a living organism we research on a particular thing, that is in vivo. But in vitro means research done in a laboratory. Okay, in test tube, in petri dish, etc. Okay, so applying FISH technique, that is fluorescence in situ hybridization technique, in vivo DNA or gene expression can be done. Okay. Then FRET. Okay. FRET, fluorescence resonance energy transfer. Okay. By this technique, we can identify the protein protein interaction. We can analyze the protein protein interaction. Okay. There are many protein interaction, DNA protein interaction, RNA protein interaction. There are various types of interaction are there. Okay. But in protein protein interaction is identified by FRET technique, that is fluorescence resonance energy transfer. Then hybridoma technique. What is hybridoma? Then it's for a monoclonal antibody production. Okay. For what? For monoclonal antibody production. Hybridoma technique, ELISA. Then first of all, we know about agarose electrophoresis for determining the molecular weight. Then AFLP for parental DNA analysis. Then affinity chromatography for mRNA purification. Then cloning for foreign gene transfer. Cloning, or we can call it what? Cloning vector. Cloning vector. Okay. Okay. Then NCD, circular dichroism, secondary structure of protein. Then chromatography for protein or RNA purification. Confocal microscopy for 3D image of tissue and cell. DNA microarray for gene expression analysis. ELISA for quantitative or qualitative for identify the infection. Okay. For analysis whether it infection is caused or not. If caused, which bacteria or viruses attacked. Then FIS, okay, in vivo or gene expression. Then FRET, protein protein interaction. Then hybridoma technique, monoclonal antibody production. Okay. Then bye. I hope this video will help you. Thank you.